Alright, welcome back everyone. So now that we've finished running the anion exchange and know which fractions have our beta-gal protein, the next thing we need to do is kind of run that sample through another purification system. So this time we're going to try to run it through the size column. But to do that, we can only really load 500 microliters, and we have about 6 mLs worth of fractions. So instead what we're going to do is we're going to put those 6 mLs into this concentrating unit. So what's going to happen is I'll add the 6 mLs into this unit and then put it in a centrifuge where I can spin at 4,000 Gs. What'll happen is the water or the buffer will get pulled down, but the protein will actually kind of get stuck up here. So I'm just going to concentrate this sample until we have that 500 microliters so that we can load it onto the size column. Alright, so now I'm just about to load the protein after concentrating it to 500 microliters onto the size column. So the difference between the anion and the size is while the anion we can load 10 mLs, 15 mLs a sample, and we know that our protein will bind to the column. For the size, we have to load as little volume as possible because I don't know if you can see it, but that really long column at the back is the size column. And the way that it's going to kind of separate our proteins is by letting it slowly go down the column. So what I'm going to do now is inject our sample, as you can see in like the syringe right here. So I'm going to inject the sample. I'm just going to let it gently or slowly flow through the size column, and we'll be able to separate all of our proteins based off how big and small they are. So very quickly, what I just want to show is this is what ended up happening after I ran the concentrated samples from the anion onto the size. So the good thing about the size is that we don't really need to worry about any kind of two buffer system. We're only using one buffer to slowly push our protein. So remember, this buffer is at 150 salt, and that's really the only difference compared to the other buffers. And we push our proteins at a flow rate of about 0.5 mLs per minute, so very, very slow. So we can see that as we push our proteins, the first peak is usually aggregated protein. But as we push it, we can expect that the earlier peaks should be the biggest proteins, and the later peaks should be the smaller proteins. So if we remember looking back at our SDS page gel for the anion, the proteins were actually fairly big. So we can safely assume that when we run this SDS page, that we're going to get peaks that are actually really close together, and when you look at that, we do. The good thing is beta-gal is our biggest protein, so I'm very confident that the first peak is our beta-gal. So I'm going to just take these fractions and run them on a gel and kind of compare it to see if we actually were able to purify it. All right. So I ended up not really recording me making this gel. It's basically the same as last time. But here you can kind of see is the beta-gal sample. And after loading a little bit more just so we can get better definition, we can see that we have a ton of beta-gal. So it's looking very good that we were able to purify our beta-gal. We have some contaminants, but it might also just be a smear from the gel. But for us, this is as clean as we wanted. In the second lane, I kind of have just crude protein. So this is what we started off with. It's a very, very ugly mess. And in the end, this is what we collected. So we were able to purify all of our beta-gal from all of this junk. So with that, that's going to conclude everything for the chromatography and SDS page gel experiments. Starting, 2 weeks from now, or when we start the next lab, we will begin our BCA to try to get the concentration and figure out how much beta-gal we actually collected.